Okay, so this series of lecture videos is going to be on the peripheral nervous system. In chapter 12, we covered the central nervous system over here. So this is our nervous system flowchart. And the central nervous system was our brain or our spinal cord. When we're talking about the peripheral nervous system, we're basically talking about everything that's outside of your brain and your spinal cord. It's broken up into two major divisions. One is the sensory or afferent division, and the other one is the motor or efferent division. So basically what happens is that we have a sensory division over here. This division is we're going to sense something by, say, receptors, send the information to our central nervous system, which is our brain or spinal cord, in order for it to be processed. After we process the information we brought in from our sensory receptors, we're going to send it out along a motor pathway to have some effect on whatever effector organ is supposed to be. So when you see sensory, it's also referred to as the afferent division, and our motor is also referred to as the efferent division. So these are just terms. If we're talking about sensory being the afferent division, anything that's on an afferent pathway means that it's approaching something. So A for afferent, approaching also begins with an A. When we talk about motor, that's the efferent division. So again, anytime you hear efferent going forward, that's going to be the division that's exiting whatever the afferent one was approaching. So in this case, we're talking about the central nervous system, so your brain or your spinal cord. So sensory division is going to send information along the afferent pathway. That's going to send it to the central nervous system to be processed. Once it's processed the information is going to be sent out through the efferent pathway to go on and affect whatever target organ it's supposed to. So again, these are just terms that you're going to see over and over again, especially in AEP2. If you have an afferent blood vessel, that means it's approaching the organ. An efferent blood vessel is the one exiting the organ. So looking at our motor division, we can further break this one down. We have the somatic nervous system and the autonomic nervous system. The somatic nervous system we already talked about when we talked about skeletal muscle. So the somatic nervous system is the nervous system that innervates skeletal muscle and skeletal muscle only. When we're talking about the autonomic nervous system, that's all the other organs and tissues and vessels in our body that are going to be innervated by that. Why are there two different divisions, one for skeletal muscle and one for every other organ or tissue in our body? That's because they're broken down by whether or not we have control over these organs or not. So when we talk about the somatic nervous system or skeletal muscle, we do have voluntary control over it. So one of the definitions of the somatic nervous system, or its defining characteristics, is that it's the voluntary nervous system. We can control whenever we want to contract our skeletal muscle. Everything else, like say our bladder, our heart, our lungs, we do not have control over. We do not send a signal specifically to these organs to tell them to contract. So this division, the autonomic nervous system, is autonomic, meaning that we don't have control over it. So it's involuntary division. Our involuntary nervous system, or ANS, is further broken down into two subdivisions. And we have our sympathetic division and our parasympathetic division. The sympathetic divisions are fight or flight. And the parasympathetic divisions are rest and digest. So pretty much depending on the status of our body or whatever environment we're in, will determine which division is going to be dominating the organ at that time. So the autonomic nervous system is going to be the topic of chapter 14. In this chapter, we're really just going to focus on the difference between the sensory division and the motor efferent division just as a whole, not going into anything super specific. So again, the peripheral nervous system, that's going to be everything that's outside of our brain or spinal cord. And typically we break it down into 4 different parts. So one, when we're talking about the sensory division, we're going to be talking about receptors. So our sensory receptors will sense something, send that information along the afferent pathway to our central nervous system, process it, and again send it out our efferent pathway to our motor endings to have some sort of control. And then we're going to talk a little bit about the second part, which are the transmission lines, so how we actually get that information there, so the nerves themselves, along with if we do have some damage and there is some possibility that we can repair it, how we actually go in and repair it themselves. Part 4 in this chapter is going to be on reflex activity, which we do not cover in this course at all. So sections 13.7 through 13.9 will not be on this last lecture exam. Okay, so we're going to focus on sensory receptors in this first lecture. So what are sensory receptors? They're basically things, the receptors that can respond to changes in the environment. And these changes in the environment we call stimuli. So how do we sense that we've actually had some sort of stimulus acting at our receptors? Well, what's going to happen is we're going to have a change in membrane potential. And this change in membrane potential, if you remember, is going to be called a graded potential. And graded potentials can either be stimulatory or they can be inhibitory. So they can either depolarize the cell or hyperpolarize the cell. If they are able to reach the threshold for whatever that cell is, then they can trigger a nerve impulse. And a nerve impulse is just an action potential. Just a couple different terms. If we're talking about sensation and perception, what's the difference? Well, sensation is our actual awareness of the stimulus to even begin with, that we are actually perceiving that something is there. Perception is the interpretation of the meaning of the stimulus. So now we're processing what that stimulus is. What are we going to do about it? So all of these receptors that we have can be grouped into 3 different classifications. One's going to be based on the type of stimulus that we have. The other one's going to be based on the location. And the last one's going to be based off of the complexity of the receptor itself. So the first classification is going to be by stimulus type. So yes, you need to know these different types of receptors and what they respond to. So our first one is going to be a mechanoreceptor. So these are going to respond to touch, so something like some sort of mechanical force. They're also going to respond to pressure, and that's going to include our blood pressure, vibration, and stretch. We have our thermoreceptors that are going to be the ones that are going to be able to detect changes in temperature. But as we'll see in a little bit, they can only detect changes in temperature within a specific range. We also have photoreceptors, so these are in our special sense organs, so our eyes. So for example, our retina, and these are going to be responding to light energy. We also have chemoreceptors all over our body, and these are going to be ones that respond to chemicals themselves. So for example, when we smell something or taste something, or if we have changes in blood chemistry. So that's a huge component of AMP2 when you get to acid-base balance and being able to sense osmolality, or the concentration of different ions within the blood. Those are all going to be detected by chemoreceptors. Nociceptors, these are going to be the ones that are sensitive to pain-causing stimuli. So for example, if we have excessive pressure, sometimes inflammatory chemicals can cause us to have pain. And I mentioned previously that thermoreceptors can only detect changes of temperature within a very specific range. Outside of that range, we no longer detect a change in temperature. Instead, we detect pain. So extreme heat or extreme cold would actually be detected by our nociceptors or our pain receptors, not our thermoreceptors. Okay, the second classification is going to be by location. And classification by location falls in 3 major categories. One is exterior receptors, and these are going to be closer to or at the body surface themselves, because that's why they're called exterior receptors. So they respond to a lot of stimuli that are coming from outside of the body. A lot of these receptors are going to be in the skin and can sense things like touch, pressure, pain, and temperature. And most of these exterior receptors are going to be in our special sense organs. So, for example, vision or taste. We also have enteroreceptors, and they can also be called visceroreceptors. And all of the enteroreceptors are going to respond to stimuli coming from within the body. So exterior receptors are outside, anterior are going to be coming from within. So things like our internal viscera that we talked about, blood vessels, also chemical changes if we have any stretch in our tissue, or temperature changes within our body itself. Sometimes they can cause discomfort, but usually the person is unaware of their workings. So if we're talking about causing discomfort, something like thirst, so if we have low blood volume, that would want us to be thirsty, and that would be sensed by an anterior receptor. The third classification by location is going to be the proprioceptors, and these are also going to respond to internal stimuli just like the anterior receptors. The difference is that these are mostly connected to movement. So we're talking about responding to things like stretch that's occurring in our skeletal muscles, tendons, joints, and ligaments, as well as the connective tissue that's covering our bones and muscles. And the whole point of this is to inform our brain of the movements that are going on. The third classification is going to be by structure, but most of these receptors that we've talking about belong to more than one category. So the majority of sensory receptors will actually belong to one of two categories. So we either have simple receptors for general senses, or we have receptors for special senses. When we're talking about receptors for several special senses, these are going to be things like vision, hearing, our equilibrium, smell, and taste. And all of these receptors are going to be in the sense organs themselves. We do not cover this at all. This is covered in chapter 15. So you don't have to worry about that, other than that there are receptors that are specific for these sense organs. What we're going to talk about in this chapter are the simple receptors for our general senses. And basically, they have modified dendritic endings of these sensory neurons. So maybe if you remember from looking at the integumentary system, we saw some Meissner or Pacinian corpuscles. Those are types of receptors that can sense different things like pressures and vibrations. So that's what we're going to focus on in this chapter. So simple receptors of the general senses. Again, general senses are going to include tactile sensations, so things like touch, pressure, stretch, and vibration, as well as temperature, pain, and muscle sense. But there is no one receptor, one function relationship. So most of these receptors that we have can respond to multiple different types of stimuli. So we don't just have one receptor that can only sense vibration, one that can only sense touch. If you are sensing vibration, that same receptor is likely also going to be able to detect touch. When we classify these simple receptors by structure, the way that we group them is based off of their nerve endings. So we can either have non-encapsulated or free nerve endings, or encapsulated nerve endings. So non-encapsulated free nerve endings, most of these are going to be in our epithelial connective tissue. So they're pretty much everywhere in our body. Most of them are non-myelinated, really small diameter. So non-myelination and small diameter means that they are group C fibers. Their endings, the axon terminals, they have like these swellings on them that look like knobs. And most of these non-encapsulated free nerve ending receptors are going to respond to things like temperature, pain, or even like touch. So we've already talked about these and what their functions are previously. What I want you to understand when we talk about receptor by structure is which ones are non-encapsulated and which ones are encapsulated. And if we haven't described them previously, then you need to know what the receptor detects. So we already talked about thermoreceptors, but now I can give you a little bit more information. So we have cold receptors that are going to be activated by temperatures between 10 and 40 degrees Celsius, and our heat receptors are going to be activated between about 32 to 48 degrees Celsius. If you're looking at these numbers, basically thermoreceptors can only detect between 10 and 48 degrees Celsius, so somewhere between 50 and 104 degrees Fahrenheit. Anything outside of that range, again, is not going to be detected by a thermoreceptor. It's going to be sensed as pain. So something below 10 degrees, so something like, below 50 degrees Fahrenheit, so even 49 might be painful. And then anything above 104 degrees Fahrenheit, so something like 105, would be sensed by our nociceptors, our pain receptors. If we're talking about temperature, as you would expect, it wouldn't do us any good to detect a hot pot or a hot pan that we pick up if these receptors were internal to us. So most of these are just going to be right under our dermis of our skin. And again, we've already talked about the nociceptors. So these are going to be ones that detect pain that can be triggered by the extreme temperature changes. So again, outside of that range of about 50 to 104 degrees Fahrenheit. If we're pinched, or we can even have pain perceived from the release of chemicals. One of the specific receptors or nociceptors is a vanilloid receptor, and this is going to be a protein that's actually in the nerve membrane itself, and this is one of the major nociceptors we have. And this receptor is going to act as an ion channel, and things that trigger this channel to open are going to be things like heat, extreme heat, if we have a low pH, which means we have something very acidic, and some chemicals. So for example, Caspian is in red peppers. So when you eat something really, really spicy and you perceive it as pain, that's because of that chemical right there that's acting on a nociceptor. Also, we have itch receptors in our dermis that can be triggered by some inflammatory chemicals like histamine, and that can also be painful as well and detected by our nociceptors. The last two of our non-encapsulated free nerve endings are tactile or Merkel discs and our hair follicle receptors. So we talked about the tactile Merkel discs when we talked about or when we hit the integumentary system. I think you actually had to identify them in lab. So they were the ones that lie in the dermal papillae between the epidermis and the dermis themselves. And their whole function is to basically function as light touch receptors. We also have receptors that are attached to our hair follicles, and the endings of these nerves or the receptors are going to actually wrap around the hair follicle themselves. So we can detect light touch based off of when these hairs bend themselves. So when we feel a mosquito, we don't necessarily have our tactile or Merkel discs perceiving the light touch because it's so light. Instead, what we detect is the movement of our hairs, say on your arm. The second major category for classification by structure is going to be the encapsulated. And again, we're going to see some of the ones that we've seen previously. Your goal is to learn the new ones and understand which ones fall under the classification of being encapsulated. So what does it mean to be encapsulated? Basically, the nerve endings at the terminal is going to be covered in connective tissue, and that's why it's referred to as being encapsulated. So one of the encapsulated receptors is going to be a tactile or Meissner corpuscle, and this is a small receptor involved in discriminative touch, and it's found just below the skin, mostly in sensitive and hairless areas like your fingertips and the soles of your feet. We also have lamellar or Pacinian corpuscles, which you also had identified in the integumentary system. And these are going to be much larger receptors, and they're located a little bit deeper, so they're going to respond to deeper pressure. If we can sense the vibration down there or the pressure down there, that's going to be picked up by the lamellar or presenting corpuscle. The other ones that we have are the bulbous corpuscle, also called Ruffini endings, and these are also going to respond to deep pressure, but it has to be continuous pressure. And that's the big difference between the lamellar, which is deep pressure only when it's first applied. If we continue to have deep pressure, our lamellar or presenting corpuscles no longer sense it because they turn off, and our bulbous corpuscles are going to be the ones that are going to be able to continually sense that we have this deep pressure going on. Muscle spindles, so these are just proprioreceptors. Again, those were the ones of the body's movements that respond to muscle stretch. The fifth classification the fifth one receptor that belongs under encapsulated is your tendon organ. These are also proprioreceptors that are going to detect movement, specifically located in tendons that detect stretch. And then we have joint kinesthetic receptors, also proprioreceptors for motion, that are going to monitor specifically joint positions and motions. So when we're talking about muscle spindles, again, all three of these are proprioreceptors. Muscle spindles detect muscle stretch. Tendon organs detect tendon stretch. Joint kinesthetic receptors monitor joint position and motion. So this is a table giving you all of the encapsulated ones that we just talked about, all six, kind of showing you what they look like. This has way too much information on it. You don't need to know the specific location other than if it's listed up here. So you should know that joint kinesthetic receptors are located in joints and so on and so forth. And up here, this functional class, if it's not in the slides, you don't need to know that either.